Hi everyone. So this is Akshay Bhatt, Science and Technology Specialist, cultivated meat from India, and we'll be hosting today's swim webinar as well as informal mixer event. So let me take you all through some housekeeping items before we start. The webinar will be recorded and a copy of which will be sent to all participants after the event and will be available on our YouTube channel. Please use the chat section to introduce yourselves, highlight any relevant content, but please do not use it for any personal discussions. The presentation will be for 40 minutes long and will be followed by a Q&A session where speakers will take your questions after the presentation. So please ask your questions using Q&A button below. Renee Bell, GFI Senior Science Content Specialist, will be lending support in this webinar and this event as well, and we'll be able to respond to any general queries or questions that our audience may have today. Lastly, the webinar will be followed by an informal networking event on Meetaway for interested attendees, where you can network with cultivated seafood experts based on common interests. So please join the networking session through the sign-up link, which you all must have received in your email inboxes. Renee will also be right away dropping the link in the chat section as well. So GFI's work is completely powered by philanthropy. So if there are any attendees who come from our donor family, thank you for your kind support and your belief in us and being an important part of our journey. If you are not yet subscribed to our channel, please do so right after this webinar to keep yourself updated on cutting edge sectoral developments through exciting webinars as well as seminars. So again, no spams promised. Please subscribe to our quarterly newsletter as well, Turning the Tide, if you haven't yet, to catch up on updates, all things cultivated seafood. So it's now time to introduce the speakers for today. We are hosting 3 experts in fish cell culture who will discuss how they're applying learnings from their previous research to cultivated seafood. So firstly, we have Dr. Mukunda Goswami. Dr. Goswami is a principal scientist at the ICAR-Central Institute of Fisheries Education, Mumbai, India, and is one of the first GFI grantees from India 2021. Dr. Goswami has more than 20 years of experience in fisheries research and has led to the establishment of a national repository in India for fish cell lines. He's now developing and characterizing fish cell lines from several indigenous species, and previously has been working with zebrafish as a model organism. So Dr. Goswami will be speaking towards what kind of opportunities lie and challenges one might face in terms of cellular aquaculture as an alternative to conventional seafood. We also have Dr. Stephen Fisher, CEO, and Dr. Kristen Skimmer, partner who develop alternative tests for fish, utilizing fish cell lines and fish embryos at AquaTalk Solutions. They both will discuss the derivatization as well as various properties of fish cell lines and how to basically assess chemical risk, water quality, and improve aquaculture and fish health. It's a pleasure having you all join us today. So thank you very much. Now I invite Dr. Goswami to present first, which will be followed by Dr. Stephens as well as Dr. Christian's presentations. So over to you, Dr. Goswami.
Good morning and good evening. First of all, I would like to thank CFI for giving the opportunity to give a talk and interact with the global community. Before I'm going to the subject, I would like to talk about my organization. Can you please the next slide? Yeah, it's Indian Council of Agricultural Research. It's one of the largest research and education organizations in the world, with 101 fishery ICAR institutes and 71 agricultural universities, where more than 6,000 agricultural scientists are working. And this is under the Department of Agriculture and Research and Education, and which belongs to the Ministry of Agriculture and Farmers Welfare, Government of India. It has 7 divisions, important 7 divisions, and then out of that, fisheries science is one of the most important divisions which has been contributing to the nation in terms of education and research. Next slide, please. Yeah, I would like to briefly talk about my institute and the university where I am working. This is the first fisheries university, and it's called deemed to be university under ICAR, Indian Council of Agricultural Research, which has been contributing towards development of human resource in terms of masters and PhD, and our alumni are spreading all over the world, US, Australia, Canada, Norway, in the many other places. And then this university has 6 important divisions. Out of this, fish genetics and biotechnology is the division where I am working. And this is the organization who is going to take lead in fisheries science and research in the world. And we are looking collaboration to develop this institute as one of the world-class institute. Next slide, please. I'm briefly explaining about myself. I have been working in this area more than 20 years, starting from my days as a student, then afterwards working in different projects, then working as a faculty in genetics and biotechnology divisions. I completed 4 important projects in the fish cell line development and characterization, including development of one national repository for fish cell line, where more than 50 cell lines are being maintained. And then recently, I have got the opportunity to take up the research for cellular aquaculture, for which I'm thankful to ZFI, and I'm also thankful to Virginia Polytechnic Institute and State University Group, USA. So I'll be talking about the project in details later on. The next slide, please. You might be knowing about Indian aquatic biodiversity. We are very rich. We are having around 3,535 species, the fish species. In addition to that, we have the resources of crustaceans, mollusks, echinoderms, and seaweeds. So that's what we have, a huge aquatic biodiversity in India. And these resources may be tapped for providing food and nutrition to the community. Next slide, please. The fish in the global scenario, if you know that the aquaculture has been growing very fast, and this is one of the fastest growing food sectors in the world. If you see here in the slide, we go back to around 2000. So then mostly the fish production is coming from capture fisheries, means open water fisheries. And during those days, aquaculture played only maybe a minor role. But if you see around 2018 and 20, where it is just, you can see that aquaculture is almost equal to the open water and capture fisheries. So this aquaculture will be providing food to the global community, maybe 9 billion people by 2050, and then where 70% of their animal intake is coming from fish. But you know that there are many challenges in conventional aquaculture. So that's where this cellular aquaculture or cell-based seafood is going to play important role. Next slide, please. So when I started my cell culture work, so I didn't know about this cultivated seafood. And those days I have been developing and characterizing cell lines from different tissue samples. And then all those sheer cell lines have been used for various applications. So if you go to the next slide, you can view this, some of the cell lines we are maintaining, we are utilizing for in vitro research, and we are cryopreserving. And then maybe next slide, in a repository where I worked in my previous organization, I moved to another place in the university, which is in Mumbai. So I developed one repository where more than 50 fish cell lines are being maintained. But here in Mumbai, now my focus will be only for muscle cell line, where I would like to develop a repository for muscle cell line. So I'll discuss about that in my next slide. So these are some glimpse of cell culture system I developed, including swim bladder, eye, fin explains. Maybe next slide. So I'm quickly going through, because these are not the cell lines for which you are attending this meeting today, but I would like to tell about some of important cell lines developed for maybe food fish, sport fish, ornamental fish. Please quickly go through those slides. Yeah, maybe more than around 20 fish species, including embryonic stem cells from Leviorhita, have been developed in our laboratory, and those have been characterized. So next slide, please. So these embryonic stem cells will be a good source for developing cultivated seafood, because in my project, which I'll be talking in details, is going to develop the cultivated seafood for this important species. This is one of the most important carp species in India, which contributes more than 80% to the carp culture produced in India through aquaculture. Next slide. So I use all those techniques, Sanders techniques, for characterization of cell lines. Maybe if I am going to develop a mussel cell line, to maintain in the repository, I need to characterize using all those methods. Those methods you are using for contamination, but fortunately, none of my cell lines have this mycoplasma contamination, but I tested with data kits. Then molecular characterization for authentication, I use DNA barcodes because I have expertise in DNA barcode. Then chromosome analysis, and most of the cell lines I developed from my laboratories are continuous cell lines. Cell cycle analysis, the immunocytochemistry to confirm the morphology of the cells, their cell doubling time, cell plating efficiency, and transfection efficiency. Whether this will be going to be useful for transgenic studies. Then growth studies, which is also going to play important role while we'll be developing mussel cell line, because you know the whatever materials, reagents, medium we are going to use for developing a mussel cell culture, that has to be economic, cost friendly. That is cost effective approach we need to use. So where we do this growth studies to minimize the concentration of serum, minimize the concentration of medium, and putting more stuffs which can be exploited from fish, like mussel extracts sometimes, maybe sometimes and serum, which is because normally we are using FBS, but in place of FBS, we like to use the serum, which is derived from the specific fish and which gives better growth. And proteomic analysis. So this is the area we are also developing expertise. If you can go to the next slide. So these are some of the oligo-2 mussel cell line we developed, we characterize for confirming the cell's morphology. Next slide. So these are some of the slides we use for DNA barcoding, then chromosome analysis. Next slide, please. This is for in vitro expression to see what kind of gene of interest is expressing in your, whether we can use or not. But again, one of my PhD student is going to standardize. So in place of lipofectamine, you know very well, in case of fish cell line, the transfection efficiency is very low. So we are going to develop one synthetic peptide, which is going to increase the transfection efficiency in fish cell line. Next slide, please. Maybe toxicity studies also, we use those cell lines. Next, for virus isolation studies, we recently used the fish cell lines for nervous necrosis virus isolation and to check their susceptibility. Next here, protein expression analysis also. We are using these techniques, but if you go to the next slide, we are using some advanced techniques. It is because of our one collaborator from Indian Institute of Technology, Bombay. He has a very good facility for proteomic characterization, and in our project also we'll be utilizing his expertise. And we already for this Leviorhita, we have developed enormous amount of proteomic information in terms of peptide atlas or in terms of web-based proteome map And recently in the Journal of Proteome Research, in the cover page, they have highlighted our work. Next. So application of fish cell line in aquaculture, already I have explained about whatever characterization I did. And after characterization, I'm utilizing those cells which are good for responding to the toxicants. Those are for expressing gene, or maybe other thing I'm applying. But today I'll be giving a detailed talk about how these muscle cells, how these cells are going to be utilized for cellular aquaculture. Next slide, please. So if you want to know about more applications of this one, it's a recent review article. You can please go through this review article. Next slide, please. The cellular aquaculture, the prospects and challenges. So as I said, in conventional aquaculture, we have number of challenges. To get rid of all those challenges, we are going to use the cell-based approach. In 2003, the first time it was reported that we can produce edible muscle protein for space travelers, and that was supported by NASA. So from that, we got the clue so that we can use those cells for producing cultivated meat. So this is going to be a climate resilient production system, because you know there are many challenges are coming from climate change. But when we are going to take up this, is a very, very challenging one, because we need to understand the in vitro myogenesis, in vitro muscle structure, how the muscles are growing, and then out of these muscle cells, how they are going to contribute your ultimate cultivated meat. So that we need to explore that. The next slide, please. So if you see, in case of mammals, the models are available to study all about this muscle growth and differentiation. But in case of fish, there are muscle cell lines, but none of these muscle cell lines have been used for cultivated meat production. So now the next, so now our approach to use two ways. One is to use some model species like zebrafish, which is the article available in 2020. So we can learn a lot from zebrafish, and then whatever insights we'll be getting from zebrafish, we can apply for other food species. Keeping this in mind, next slide, please. We developed this muscle cell line from zebrafish, but those days I didn't think that these muscle cells can be used for cultivated seafood production. That was used for only some kind of gene expression studies, or the myostatin, how it is going to negatively affect the muscle growth, all those. But those days I didn't pay attention from this point of view, cultivated seafood production point of view. But now I have realized whatever knowledge I have gained from this muscle, as I showed in the previous slide. So this is going to give the root to understand myogenesis in a very effective and a very timely manner. So it will take less time. We can understand this so that we can apply those knowledge gaining from this zebrafish model. Next slide, please. So here I am going to thank GFI, Good Food Institute, for giving me the opportunity. Just I am taking 1 minute to tell about my association with Good Food Institute. Those days when I developed cell lines, as you have seen, then the cell lines have been developed from many various tissue samples, maybe 1 or 2 from muscle. Then the thought came to my mind, and I wrote to Professor Mark Post, who developed the cultivated meat for the first time. Then he nicely emailed and talk about GFI, and even GFI India can help me in this regard. Then immediately I contact Dr. Desmukh from GFI India, and then Dr. Claire. Then afterwards, my journey has started with the GFI and has been consistently supporting me, encouraging me to take part in this competition, the proposal, and I participated. And I am thankful to my collaborators from VT USA, Virginia Tech, Dr. Reza and Dr. Laxi. And then we have got this proposal. And then as Dr. Akshay said, this is the first proposal. So we have many challenges. And then after this, so my focus, whatever I have been working, whatever I have been using my cells, I have diverted my focus towards cultivated seafood production. The type of fish muscle, mostly we know white meat, white one, red one, and maybe in between headed, white and red, maybe little bit pink one. So here again, some lean fish, fatty fish are there. This Leviorita belongs to the Cyprinidae family. As I mentioned earlier, the zebrafish can give us more insights, because maybe in this fish we are getting the first growth of the muscle cells within maybe 1.5 month space. We are getting 18 pulses, and then again this fish is a lean fish. So this will be quite interesting to know how this myogenesis is happening, and maybe we'll be doing some studies to understand their molecular mechanism which is behind this. Maybe it's in editing CRISPR techniques also in the future. We are thinking to understand and then proliferate and then speed up these cells, and maybe at the right type of cells we can get at the end. Next slide, please. The techniques for cell-based meat production, I think you all are aware of these techniques. So here the source, as I am telling, the myosatellite cells, as I have been getting the cells, and the cells are growing very fast. By now, my second plan will be to isolate myosatellite cells, and then how these myosatellite cells are going to differentiate into the myocytes or maybe myotubes, or then if this, my collaborator will come up with some right kind of scaffolds, so I can see then how these cells will be good enough or the cells are not good enough to get the ultimate seafood meat. The next slide, please. So again, organ printing, maybe using some kind of biomaterials from marine resources. Again, it will be a good option. Maybe you might have seen this paper. So we can utilize more marine resources for ultimate, see the structure of this meat before going to the market. Next slide, please. So this is way forward. So based on my experience over the years and my recent experience from this muscle cell culture development, so what we need to take as an urgent requirement, or say the need of the hour, the species selection, which species we are going to take, what cell types will be ideally useful for ultimate cultivated seafood production, whether this will be a market driven or whether we'll be taking some model species, and then understanding of in vitro myogenesis and adipogenesis, and then maybe your co-culture of both, because if you are going to get the original flavor and taste of fish in the cultivated seafood, so we need to work out this. And maybe along with my proteomic characterization, we are going to get some kind of proteins which will be helping to get original taste and flavor, because without this original taste and flavor, our mission will not be successful. Then optimization of appropriate development of appropriate muscle cell lines, and then optimization of those muscle cells where we can grow, maybe using serum premedia or maybe some other growth factors. As I said, maybe some growth factors or some kind of stimulating factors we can get it from the fish itself, like muscle extracts or something serums, or maybe some food grade reagents. And then how these are going to differentiate, and some differentiation medium, because I will be requiring this collaboration, or I will be requiring this help from the people, those who are engaged in this cultivated seafood meat, then scaffolding, then bioreactor design, because ultimately we need to have large number of cells. It's not the laboratory we are getting in the flask, whatever cells we are getting, that will not be good enough. And then culinary effect, by understanding these proteins, by understanding their differentiation, and then what important role is going to play is in networking and collaboration. Because so this is and it's not in the isolation, means only doing culturing the muscle cells will not be enough. The food technologist, the chemist should come together. That's what I have been telling my CFI India. So whatever nearby institutes are there, they have expertise in chemistry, understanding more about this myogenesis, and in applying those knowledge and then expertise in fish cells, and all together in the form of a networking. Maybe in India, like now in US, they have this Institute for Cellular Agriculture. My vision is to develop a National Institute for Cellular Aquaculture in India, where this CFI is going to play important role. And then whatever cell lines I'll be developing from mussels, so I'll be maintaining in the repository and focusing one cell line, maybe taking help from model, and then whatever other cell lines will be there also will be maintained for the scientific community. So this is what the sustainable approach is required. Means I am getting a grant maybe for 2 years, I'll be developing cell line, I'll be characterizing. It will not end here. That has to go long way to the ultimate cultivated seafood production. This was published in the Nature in 2019. And what is the message here? Academic research can play a significant and lasting role in taking this research to the market. Taking the research to a market means your ultimate cultivated seafood. So without this academic research, it is not going to be possible. Next slide. Just I would like to acknowledge the Director General, Deputy Director General Fisheries, Indian Council of Agricultural Research, New Delhi, who has supported maybe within a maybe 2 months, it takes 1 year, 2 years. Maybe they're realizing the importance of the topic. So they have given me the approval. I would like to thank my mentor, W.S. Lakra, who taught me the cell and life together. And then I started working with his guidance. And today it's fortunate, the dynamic and a very supportive director has joined recently, Dr. Ravi Shankar C.N., who has been supporting, and then he has given me the cell culture facility lab to take this work forward. So with this group of students down here, you can see, so I have a big group and I can contribute, and I am waiting for the day that from India, the first lab fish meat seafood will be produced. Then I can contribute for that, then I'll be very happy. And then, of course, and during my previous work, Director ICR NBFGR Lucknow, then DVT, who has been instrumental in supporting projects for development and characterization of cell lines and maybe developing repository, ZFI USA and then ZFI India group, Professor Sanjiva, who has been helping me for proteomic characterization. So that's a very strong hand we have. And then Dr. Reza and Dr. Lexi from BT USA for their kind collaboration. And my students at my previous institute and my present institute. Thank you so much. Thank you so much for giving me the opportunity, for giving this grant for this work. Thank you so much.
Yeah, thank you, Dr. Goswami, for sharing your knowledge with all of us and giving insights into your cultivated seafood project and highlighting some of the challenges which you have been facing and the progress which you have made so far. But without further ado, I'd like to next invite Dr. Stephen as well as Dr. Kristen with their presentation, and after that we'll go for the Q&As. Thank you.
Thank you very much. And hi, everyone. While Stefan is getting set up, I can maybe already briefly introduce ourselves. Okay, so I think we are all ready to go. So we are very grateful to the Good Food Institute for giving us this opportunity to present here. I would like to start with a little bit of a disclaimer. We are not, maybe yet, involved in cellular aquaculture, but we think it's a really exciting field, and we share the mission of the Good Food Institute. And so hopefully we can also contribute with our expertise, which also lies in fish cell line development as animal-free and resource-efficient alternatives to fish. You will see it's a little bit a different background that we have, which is mostly focused on fish physiology and toxicology. And I share this presentation with my colleague Stefan Fischer, who is the CEO of AquaTalk Solutions, which is a spin-off of ERWAG, which is the Swiss Federal Institute of Aquatic Science and Technology, where my group is located. Now, I'm sorry if I take you almost a little bit a step back, but I think it's still very useful because we basically wanted to share what we mean by a cell line. So we talk about fish cell lines. So we talk about a population of cells from a multicellular organism, in our case, fish, that can be propagated outside of the organism through serial transfer from one fabricated culture vessel to another. Now, I just wanted you to know that it's quite interesting to realize that when the first human cell line was derived, the famous HeLa cell line, was exactly the time when also the first fish cell line was derived from rainbow trout, the so-called rainbow trout gonad cell line. So the origin of these vertebrate cells was from the timing here very similar. Now, how is a fish cell line born? So you've heard about a lot of cell lines that have been developed by our previous speaker. I just wanted to show you a little bit how it actually works. So you start with the fish that you're interested in, and obviously you can also do this from fish species which are hard to culture or also threatened species, and you isolate the tissue that you're interested in. Now you essentially have two ways to process these. So either you cut small tissue pieces, they should be no larger than 1 cubic millimeter, or you can use enzymatic digestion to obtain a cell suspension. Now I'm showing you here two examples. On top for tissue pieces, actually you see gill fragments, which actually led to the development of a very famous rainbow trout cell line, the RT gill W1, and you will hear more from it later. In the bottom figure, you see how liver cells behave when they have gone through an enzymatic digestion to obtain a cell suspension. So they typically form these ridges. Now, with a lot of nurturing, the right environment, and also quite some experience, you may succeed into cells growing out of these initial preparations. And as soon as you have enough cells that you can basically propagate them in a new culture vessel, you have a cell line. Now, obviously, there are two things that can happen with your, cell line that you're so proud that you have managed to get. If you're unlucky, the cell line after some passages may stop proliferating, in which case you have a limited cell line. This can still be very useful if you can freeze down, several passages, early passages. It can still be very helpful to have a limited cell line. Now, however, you may also be able to propagate these cells essentially indefinitely to obtain an unlimited cell line. Now, my point that I would like to make is that for fish, at least from the experience that we have, they easily immortalize spontaneously. So what does it mean? So they take this immortal status without us interfering in any way, no genetic manipulation or anything, which also means that we can derive these cell cultures from healthy tissue. Now, why fish cells can do this is not yet fully explained, but there are two hypotheses. One is that even if we take adult tissue, we isolate stem cells along. And the other one is that fish have a high telomerase activity. And I would like to show you two lines of evidence to support both these hypotheses. So the first one is on stem cells. Together with colleagues from Italy, we have actually characterized 3 rainbow trout intestinal cell lines obtained from adult rainbow trout. And what you can see here is that, first of all 3 express on a gene expression level, so mRNA, typical stem cell markers. This is what you see on the left side. Now, the red and blue bars show you that if you use immunochemistry, you also find a high percentage of cells that have stem cell markers. And the percentage decreases with higher cell density, which is what you would expect. Now, in terms of telomerase activity, the fish cells that we have are known to have such a high telomerase activity, which you in mammalian cells only find in cancer cells. And I show you here on the left side 3 rainbow trout cell lines, so denoted RT. And you see the telomerase activity in comparison to a mammalian cell line, the so-called Hoover cell line as an example, which is a humpback whale cell line that we have also established. And you can see that its telomerase activity is only as high as in the fish cells if we purposefully transfected it with telomerase. So this would be the black bar. Now, since the first cell line, the RTG2, was established, the number of cell lines has increased dramatically, or quite a bit, not as much as with mammalian cell lines, but we've heard about this also before. A very nice research to look into which cell lines are out there is the Cellosaurus, which also lists fish cell lines. And just before Christmas, the people who launched this platform reported in a tweet that they have now sort of recorded the 800th fish cell line. We have been mostly focusing on rainbow trout. We are also developing cell lines from other species, but here is to illustrate the portfolio of cell lines that we have from rainbow trout. And so we use them to understand more about fish physiology, also toxicology. We also develop new pipelines. For example, it was mentioned before that fish cells are very hard to transfect. So we established a CRISPR-Cas pipeline, which includes very high transfection efficiencies. And we use these cell lines for toxicology. And so here I would like to come back to our star, the so-called RT-GIL-W1 cell line, of which I showed you a very early preparation earlier. We have actually used the cell line to develop an assay which allows us to predict acute fish toxicity. Now, this is very important because it causes millions of fish used and killed in such experiments worldwide today to analyze water quality or also chemical toxicity, as well as I will say that this assay is extremely helpful to also monitor water quality in aquaculture. Now, we have really worked on this assay from conception to international recognition. So this was a 13-year process, I can tell you. And we have now this assay established as an ISO standard for water quality testing, as well as an OECD test guideline for chemical toxicity assessment. And so with this, I would like to hand over to my colleague Stefan, because the standard and guideline are a core expertise that we offer in AquaTalk Solutions. And he will also guide you further to how we can use cell line-based assays to monitor fish health. So with this, Stefan, I hand over to you.
Thanks, Christine, and hello, everyone. Here's Stefan Fischer from AquaTalk Solutions. As Christine has explained, we are a spin-off of ERWAC, the Water Research Institute from the ETH in Switzerland. And it's now a pleasure for me also to show you some examples how this fish cell lines can be used as alternative test models for environmental risk assessment and other approaches. For the prediction of acute fish toxicity, as Christine already explained, according to this ISO guideline or OECD guideline, we use this RT-GIL-W1 cell line. So it's a GIL cell line And you may ask yourself why we use gill saline. So the gill epithelium reflects the primary contact and absorption point of pollutants, because fish are permanently filtering water through their gills, and the effect on gills could cause lethal effects. That's the reason we use the gill saline. And then this gill saline is seeded on a 24 well plate and exposed to the test chemical, dilution series of the test chemical or of the water sample. And then it gets exposed for 24 hours. And after this 24 hours of exposure, the cell viability is determined by using a combination of 3 fluorescent indicator dyes, which are measured in a fluorescent plate reader. Based on the with this, we can obtain lethal and sublethal effects, which detect or the lethal and sublethal effects are detected through a reduced signal of this indicator dyes. And here we can obtain different cellular responses like lysosome integrity, metabolic activity, and membrane integrity. And based on this outcome, we can also then calculate effect values like EC50, NOAC, LOAC, non-toxic concentrations. And furthermore, there are different validation studies which has really proven the how good the saline assay is. So, and how good it correlates with the standard acute fish toxicity studies, so with juvenile or adult fish. And as you can see here on this both graphs, so there are both studies compared different amount of compounds with different chemical and physical properties. So as you can see there, the results are really close together. They really rely on one line of unity, and it really shows that the outcome is almost identical. So it's a really good proof how suitable such saline assay is to predict acute toxicity to fish. And then we are not only measuring acute toxicity, we can also detect sublethal effects or can make prediction on chronic effects on fish. And for the sublethal effects, we analyze via gene expression analysis of biomarker genes at different endpoints. And why we do this? So because the first interaction with any kind of stressor or chemical happens on the molecular level. And the first unit which is regulated there is the so-called mRNA transcription. And based on this, we can obtain different cellular responses like endocrine disruption, immune regulation, biotransformation. So almost every kind of cellular response. And based for this, especially for the trout salines, we have established really a huge variety of different markers, which are really specifically established and validated. And for the chronic effects, we can make some prediction on fish growth. So by measuring cell population growth. And you have to assume, so this really, it's for a standard fish test, according to OECD, such test is a really long and really expensive test. And based on using the cell line, we can really reduce this and we will get the same outcome. So this was also, there is a study which we can also share later, which really shows that it has really extremely good correlation. And we are using this fish salines not only as an alternative for environmental risk assessment, they can also be used to be really helpful for aquaculture. As you all know, this is also one reason why we are here nowadays, more than 50% of the fish that we are consuming derives from aquaculture. And there, millions of fish are yearly used for so-called feeding trials to test new fish food additives or fish drugs to improve fish life conditions and so on. And we have very well established intestinal rainbow trout cell line, so-called the RT-GUT GC, and it's perfectly characterized, so we know everything, and it's a really ideal model to test effects on fish health. And I don't want to go into so much into detail, but I want to show you what we are able to measure. You can see here on the left side the in vivo situation, so what it be, how it looks in the gut, in the fish gut. So we have the intestinal lumen, the gut epithelia, and the blood-facing side. And with the gut cells, with our setup, we can also mimic a similar setup in vitro. So we have this transpose system here. In this insert, the gut cells are growing as a monolayer, and this insert has a semi-permeable membrane. Then we can mimic the intestinal lumen and the blood-facing lumen. So what happens? You all know a fish is taking up food, it enters the gut. What happens there? So food gets digested by enzymes, digestive enzymes. And we can also do analyze and measure this in these gut cells. So the gut cells are also producing this digestive enzymes, and we can obtain then this digestive activity. So how good is food digested? Another thing is uptake of compounds, either if they are digested or we will not digest it. So to see how compounds are taken up from the intestinal lumen over this epithelial barrier into the blood-facing side. And this uptake of compounds we can also directly measure in our system. Then something else, you know, why either physically or maybe also due to effects of toxicants, this epithelial layer in the gut could be somehow disturbed or damaged. And what would cause, or what would be the result of this is that, for example, compounds can easily enter the blood-facing side, because when this barrier function is not existing anymore. And therefore, it's also important to know to measure the epithelial tightness or integrity. And we can also do this with our setup. So here we use electrodes, so which we place in the apical chamber and in the basolateral chamber. And then we can measure the electric resistance. And if there is a high resistance, it means the tightness of this epithelial layer is pretty good. So an integrity is also good. If the resistance is going down, it means somehow this epithelial layer is effective. So, and as you can see here, I told you if this epithelial layer is damaged, but it's damaged, the organism also reacts to this. So we have some healing processes, or proliferation processes are ongoing. So that means body reacts and then tries to heal this layer, for example. We can also mimic this quite well. We have a so-called cell proliferation or wound healing assay with a bit of different setup. We are using here such a tubal insert where we grow the cells in these two chambers. Then we can open this and then we have a gap. And then we can monitoring over time how fast the cells proliferate into this, into this gap or close this gap. And then we get already an idea about wound healing or also growth. Then, we also offer a so-called immune challenger assay, where we can assess effects on diseased fish. And what we can do, we can mimic bacterial, also viral infection. And this is also for aquaculture. It's really important because there they use a lot of fish drugs or antibiotics to avoid fish disease. And then also a lot of companies are specialized to produce additives which are stimulating or have a good effect on the immune system. And here I will show you one example of one of our clients they produce food additives, and which should have a good effect on the fish immune system. And we got blind samples, and we did this challenger assay where we stimulated bacterial infection with lipopolysaccharides from E. coli. And you can see here, this is the control without the cells, where the immune system is stimulated, is upregulated due to this LPS exposure. And then we did also a co-exposure with this blind samples, and we have a positive effect if this upregulation is going down. And you can see we've identified 2 candidates where we saw significant differences to the control. And this client also did further afterwards a feeding trial, and the outcome was the same. Therefore, I have to say, you know, sorry, that this gut cell line is really helpful also for aquaculture to reduce fish feeding trials, to really test fish food additives or fish drugs. With this, I will hand over to my colleague Christine again. Thanks.
Okay, thank you very much, Stefan. So after these examples of where we employ the fish cell lines for aquaculture and also toxicology, I would like to point out a few maybe take-home messages for using such fish cell lines for cell factories from our novices' point of view. So first of all, I think fish cell lines really offer the opportunity to really develop many different cell lines from different species and different tissues. And I think the ability of fish cells to spontaneously immortalize from healthy tissue is an extremely valuable feature. Now, I've shown you that we also find stem cells in cultures that were starting from adult fish. And so I think there's great potential also in isolating these stem cells to then use them for differentiation. And my final point actually is one of the things that has not been mentioned this afternoon is a severe bottleneck from our point of view for using these cells in cell factories. And this is that there is so far no routine fish cell culture medium that is animal component free. So here I wanted to point out to you that we do have a project running at ERWAG, which is headed by Dr. Barbara Joseph, to develop an animal component-free fish cell culture medium. And she has very much advanced to having a serum-free medium as a start, so fully defined serum-free medium for routine culture of at least the rainbow trout cells as a starter. And so with this, we are at the end of our little presentation, and we would be very happy to answer any questions. Thank you.
Yeah, thank you, Stephen and Christian, I think for the wonderful presentation. So also for throwing light on the importance of, let's say, testing, for example, for infections or toxicity in fish cell lines, just like how it's actually done, regular, normal, conventional aquaculture is important. And in the long run, would probably lend weightage towards, let's say, easing regulatory hurdles or roadblocks, and obviously creating animal-free media formulations has been one of the biggest bottlenecks we have been pushing for researchers to work on, or companies to work on, and creating those for the future of this sector. So thank you for throwing a light on all of these, and now we are open to Q&A. So I think in the interest of time, most of the questions have been answered so far, but there are a few questions which probably we can take live before we move into our networking session, which is going to start in about, I think, a minute or so. Yeah, do you have, I think this is all for Dr. Goswami probably. Do you have, I mean, others, I mean, Dr. Christian, Dr. Stephen, please feel free to answer as well. So do you have any data showing myoblast differentiation to muscle? If so, the differentiation is, how's the differentiation turn out after several passages of cells? Dr. Goswami, if you can just answer this.
So actually we are doing this test from the day one. We are getting the when cells are radiating from the explant, and then cells are growing and differentiating. We are going to study this, how the differentiation is going to place, and what stage we are going to get, maybe after how many hours we are getting the myocytes or myotubes or myoblasts. So that needs to be standardized. That needs to be investigated.
Got it. Thank you. And is the stemness of fish satellite cells easily lost after serial passaging? Do you know about this, Dr. Goswami? Basically, how does the stemness work in terms of, let's say, after several passages?
Actually, we are going to standardize this for a myosatellite isolation. We haven't done so far. We don't have experience with this one, but it gradually declines. Maybe you are what kind of fish you are using, what kind of cells you are using, that is going to affect this differentiation, the declination of this differentiation.
Got it. And there is one last question: where can labs purchase fish myoblast cell lines? I think that's the biggest bottleneck for us right now. I think we don't have that. That's why collaborations are very important to push this field forward in terms of development of cell lines. We have a couple of initiatives within GFI itself. That's obviously a white space idea, and this is one of our high priority projects, basically focused on developing and increasing access to cell lines from aquatic species. And we also have a cell line repository where people who have or creating or are in the process of creating cultivated fish or cultivated seafood cell lines, please visit our website, and we can you can just deposit your cell line there as well. Yeah, I think most of the questions are done. And now it's time for a networking session. Claire from our team has mentioned that she can still let in people after a couple of minutes. So please feel free to join the networking session and meet experts and like-minded people and discuss about cultivated seafood. So thank you once again, everyone, for participating in this webinar. And thank you, speakers, for making time for this webinar. And see most of you in the networking session. Hope you have a good time there. Thank you, everyone, and have a good evening, good morning. Yeah.